Right. So welcome back, everyone, to the sort of second part in this guest lecture series on single-cell RNA-seq analysis. And so today I will pretty much start off or pick it up where we left yesterday. So I'll first be talking about, Tuesday, where we're talking about how to do some dimensionality reduction, feature selection, and visualization of your single-cell RNA-seq data. So first of all, something you may have heard of, because it's something which is not just for single-cell RNA-seq, but pretty much any other types of data where you have really high dimensions. This thing called cursive dimensionality. So very loosely speaking, what that means is basically that when you get into very high dimensional space, sort of distances, be it Euclidean or really any other type of distance that you want to consider between data points, sort of become less informative. So what that means in practice is that sort of pretty much wherever you are, every other data points look to be sort of more or less at the same distance. So distances are no longer as useful as they would be in lower dimensional space for sort of discrimination and other tasks. So one example is sort of shown here in this plot here. So here we took a small data set on early mouse embryonic development. And we consider these sort of two different scenarios where you either use sort of all 20,000 genes that were profiled in the samples, or you sort of use a selection of the most variable genes, only 500 of them. And as you can sort of see here visually, you have both sort of the clusters are sort of more distinct here on the left compared to the right. And you can also see this sort of quantitatively here, that the fraction of variance explained by the different principal components is higher here. So, so this reduction of the number of features has sort of allowed us to sort of better pick out the sort of the biologically meaningful structure in this data set. So this is sort of a very useful strategy for dealing with this problem of the cursive dimensionality. So this is a sort of example of what's called feature selection. So by feature, we mean gene here, really. So the idea is to sort of not consider all of the genes in your sample, but really only a subset. So one of the things that you always should do is to sort of just look at for the genes that are just really lowly represented. Sort of typically when you would use this sort of a threshold of a few percent. So if a gene is present in no more than a few percent of the cells, then you just take it out altogether. So that can typically reduce the number of genes by sort of a few thousand. It really depends on how deep the sequence is. But then you want to go sort of further than that. And one strategy, which is the most popular one in the field, is to look at sort of highly variable genes. So it is, of course, the case that the variance is proportional to the mean. So we can't just look at the variance per se, but instead we look at the normalized variance of a coefficient of variation, which is sort of shown here. And so here's a sort of a plot from where this method was published, which, as you may see, is actually very long ago by single-cell RNA-seq standards. So what you can see here, each dot represents a gene, and then you look at the mean expression versus the normalized variance. And we're interested in the genes that have sort of so the red line here represents the sort of the mean, the average trend, and we're interested in the genes that are higher variance than you would expect by this trend. These are the pink ones that show up here. And so what's worth pointing out here is that the blue dots correspond to these ERCC spike-ins. So these are sort of artificial spike-ins that have been added to your sample. So spike-ins is really something that was sort of very popular in the sort of Jurassic era of the single-cell RNA-seq field, which would sort of correspond to like 2014 or 2015 or so. So you may have to ask a sort of senior postdoc in your lab what they are and why they're useful, but they've really sort of fallen out of favor these days for various reasons. Hence, I'm not really talking about them. But these are supposed to be sort of negative controls, and as you can see here, none of them are highly variable, which is a nice thing, a nice confirmation. Another strategy is to, instead of focusing on the sort of the genes that have high variance, is to focus on the ones that have high levels of dropouts, so more zeros than you would expect. So the trend that you'll see in sort of pretty much every data set that I'm aware of is that you have a very strong correlation between the dropout rate, so the fraction of zeros, and the average expression. So what that means is that if the gene is highly expressed, you have few dropouts, and vice versa. And you can sort of plot this, and you can sort of fit this type of sigmoidal curve here. And then what we're interested in are the genes that are sort of illustrated here in purple. So these are the dots that are sort of have a higher number of dropouts than you would expect by chance. So those we can pick out and use as features instead of the highly variable ones. These will to some extent overlap, but they're not the same as the highly variable genes. So another strategy that we can use to deal with the curse of dimensionality is called dimensionality reduction, which is a very big field in itself. And there's been lots of work done on that in statistics and mathematics, and lots of methods that have been applied to single-cell RNA-seq. And the reason why dimensionality reduction works here is quite straightforward, right? Because so you have 20,000 genes, but then you can imagine, for example, that you have one transcription factor which controls sort of say 50 other genes, right? Then in reality, those 50 genes don't have 50 separate dimensions, but they can describe by one dimension, which represented by this transcription factor. So this sort of more formally speaking, what we have is a so-called manifold hypothesis, which is that there is this sort of lower dimensional structure embedded in this higher dimensional space where all of these points, the data points, the cells actually live. And so what we're trying to do is identify this low dimensional manifold, and then we can use that to represent the data instead. So this is of course hard to illustrate in because you can't really draw more than 3 dimensions, but this is sort of very famous example for illustrating this. So this is called the Swiss roll. So you can see here that we have this sort of structure which sits in 3-dimensional space, but in reality it's actually a 2-dimensional surface. So ideally your method should be you only require two coordinates to describe this. So ideally your method should be able to identify that we have a 2-dimensional surface sitting in 3-dimensional space. So there are multiple methods for trying to deal with this. The most popular one by far, and is really an old workhorse from statistics, is called principal component analysis. Really, the main advantage of principal component analysis is that it's linear. There are sort of very efficient methods for calculating, even for large data sets, and it's easy to interpret what it means because the different directions that it picks out are the ones that explains the most variance. So we can easily interpret distances and also angles and positions in the PC space. So that's a big advantage of it. Fortunately, it doesn't really work that well for when you're trying to do visualization. I think the main reason is, again, sort of intuitively speaking, it can often get sort of quite dominated by sort of a few outliers. And what that will result in is that most of your data points will sort of go into one sort of small bunch. And they will sort of sit right on top of each other. So it's very hard to sort of make out any interesting structures that's happening here. So PCA will work well for really small data sets, so up to a few hundred cells. But as soon as it gets larger, you'll probably need to resort to some sort of nonlinear method to better sort of position your points from the high dimensional space into the 2-dimensional plane. So there are lots of methods around for this. What was initially the most popular one is called T-distributed stochastic neighborhood embedding, or t-SNE for short. And so it's a sort of it was developed sort of in the more general machine learning field, and it's been around for quite a while, and it's very popular, and it does its job really good in terms of sort of putting the points down into the plane. It has some issues. So one of them is the S, which is stochastic, which means that when you run it different times, you'll get different outcomes. And then a bigger problem, however, is that it has a free parameter, which is called perplexity. And the outcomes, as you can see here in this figure, is that the outcome will sort of be highly determined by how you choose the value of your perplexity parameter. So here's sort of the real structure of the data points, and here's sort of how they look after going through the t-SNE algorithms for different perplexity values. And one of the issues is that there's no procedure for optimizing or how to choose your perplexity value when you run this. So you can almost make the data really look sort of whichever way you want it to. So that's quite problematic. And it's what's very important to stress here about t-SNE is that it is really only designed for visualization. And you should really not try to do sort of anything with the output beyond sort of looking at it, and not sort of try to interpret things the way you would interpret them using a PCA. But I think, unfortunately, that is not something which is always heeded by people in the field, and you can often sort of I feel people can sort of start discussing t-SNE plots as if they were PCA plots, and which is not the right thing to do. So t-SNE has a little bit fallen out of favor in the field. There is this newer method called UMAP, which sort of pretty much does the same thing as t-SNE. It also has like a sort of it has like this approach for doing this is based on sort of graph theoretical properties or doing a graph analysis. They argue that it's better at preserving the global structure, because one of the things that these two methods do is that they tend to focus on sort of local structures and make sure that they are sort of guaranteed to be well preserved, but there's fewer guarantees about the global behavior. And UMAP claims to be doing better. I'm not 100% sure that I've seen any really strong evidence that that is the case, but again, it's what people are using mostly these days. There are other methods. Another strategy is called sort of force-directed layout or graph layout. And again, a method which goes back quite a long time, sort of several decades, been around for a long time. The idea is very simple. You sort of think of your data points as being in this graph, where each point, the pairs of points are connected by springs. And then you essentially have a mass spring network, and you sort of let that equilibrate, and that gives you the layout of your data points. So one advantage of this is that it gives you much more reproducible results. So as you can see here is that you have sort of 3 different samples from the same type of tissue, and this sort of spring method with a forced graph layout sort of gives you sort of more or less the same topology every time, whereas when you run it with t-SNE, it looks quite different every time. So that's obviously a very big advantage here. I think the drawback of this method is that it can be computationally very costly to run it. So if you have a large number of cells, this can be problematic. So I think this is the end of the first part. Stop recording. Right, so now we're getting to the sort of section where we start talking about methods that can be used to sort of give more like sort of biological insights, or for more biological analysis of your data. And the first thing we're going to be talking about is clustering. So clustering is very useful because it allows us really to get to one of these really central questions in biology, which is sort of how to define a cell type. So traditionally, sort of cell types were really defined based on the morphology, but then people sort of started to sort of finding these different types of protein surface markers that they could measure, and that allowed you to get a sort of a more molecular based definition. But then, of course, with single-cell RNA-seq, you can take that even further. Instead of just looking at a few surface markers, you can look at the entire transcriptome, and then you can have sort of like a bottom-up type of definition of what a cell type it is, and it can be defined based on the transcriptional profile. So this is, of course, sort of very attractive for trying to be a little bit more rigorous about how to define cell type, but it's of course not without problems from a computational point of view. So what this really means, computationally speaking, is that we want to do unsupervised clustering, and this is again a problem that's been around for a very long time. in the statistics and the mathematical literature, and it's quite challenging because we're just given the data, and then what we have to do is to figure out sort of how many clusters there are and where are they, where should the different points go. So there are several different strategies out there. The oldest one is k-means, which is sort of quite intuitive. So you assume that there are sort of k different centroids, and then that you try to group the points in your data set to these centroids. So this can be done computationally very efficiently using the what's called the Lloyd's algorithm. It scales quite well with the size of your data set. So that's why it's an attractive strategy. It's also sort of intuitive to understand what's going on and what the algorithm is doing and what it's finding. The method one method which is based on k-means clustering, which is used in single-cell RNA-seq, is called single-cell consensus clustering. So what this method essentially does is that it runs k-means clustering sort of multiple times using sort of slightly different input parameters to how it's doing the clustering. And then it takes all these different clusterings. So each of these different parameter combinations, you can think of it as it provides a different solution for how the cells should be grouped. And then it takes all these different solutions and it combines them into a consensus, and then it sort of gives that as its final result. So the main advantage of this is very robust, and it's also quite remarkable that the consensus that you'll find is often going to be better than any of the individual solutions that you have. So this sort of wisdom of the crowd types of approach works really well. The main drawback of this method is that it's very slow, and you can't really use it sort of reliably for data sets with more than 1,000 cells. So another sort of type of like generic clustering strategy is called hierarchical clustering. So this is not very popular in the single cell field. I think the main reason is because it's quite slow, so it scales quadratically with the number of cells that you have. One major advantage of hierarchical clustering, however, is that it doesn't give you just one solution, but it gives you multiple solutions. You can you can get this type of tree here, and you can and then once you have done that, you can sort of choose where you want to cut in the tree, and that will give you the final outcome. The type of strategy which is most popular these days are these graph-based methods. So these were sort of really emerged in the noughties and in the last decade, and they were really devised for sort of looking at these very large internet or World Wide Web related data sets. So the main advantage of these methods is that they scale very well, and they can handle sort of millions of nodes or millions of cells at a time. So the most popular method is known as the Louvain method, or Louvain clustering. And what it will do is that it tries to sort of maximize the modularity. So modularity is modularity is defined as a sort of a set of nodes that have a lot of internal connections between themselves and very few connections to the other nodes. So it tries to sort of optimize this quantity, and then it does so in a sort of hierarchical fashion. And again, the main advantage is that it scales very favorably. It's n log n, and it can be run on these really large data sets. So one big challenge of clustering is that we typically don't really have a ground truth. So it's very difficult to sort of evaluate to what extent the clustering is sort of correct. So there are sort of various heuristics that you can use to, and what they typically do is that they look at sort of how tightly are your points sitting next to the centroids or cluster centers that you had identified. So my favorite method for doing this is the silhouette score. And it can sort of, it's quite easy to interpret, and you can sort of calculate one silhouette score for your entire sample, which will tell you sort of how good the sort of the overall clustering is. But then you can also calculate for sort of the individual clusters to sort of identify which clusters are the ones that are sort of performing poorly, and where you might have be able to do a little bit better. So another important thing to say about clustering is that it's typically the case that there is not a single correct answer. So you can, for example, imagine if you have a data set from say a cortical brain region, then you could cluster that on multiple different levels. So you could sort of, for example, you could have, on the one hand, you could have like very high level clustering, so where you distinguish the sort of the neurons from the astrocytes and the microglia. And then you could imagine a sort of a more fine-grained clustering where you sort of neurons are split into the excitatory and the inhibitory neurons. And then you could think of it at an even finer level where the neurons are sort of split into the different layers where they would come from. So all of these solutions would sort of be correct in some sense, and which one is sort of best really is really determined by your question and by your application. And I think my sort of advice and recommendation is for most applications, it makes a lot of sense to sort of consider clustering at sort of two different granularities, and then you want to make sure that sort of the results that you sort of get from one granularity is sort of robust in the other one as well. So that's often a useful way of working with this. So one important sort of caveat to say about pretty much all of these methods I've been talking about before is that they tend to perform well for the large clusters, but they typically have a really hard time for very small clusters, which would of course often correspond to like rare cell types. So the reason for this is sort of intuitively, it's not hard to understand, because if you have a really small cluster, it's really hard to know if this is sort of a real sort of biological outlier or if it's just some sort of noise or sort of technical outlier. So if you know that you have or suspect that you have these sort of small clusters, so for example some type of stem cell or something, then it is often a good idea that to use a different method than the clustering method that I just talked about for trying to look at them, because there are these methods that are specifically designed for identifying these rare cell types. So one example is the race ID method, which is sort of based on k-means clustering originally, but then it has sort of special extra steps that allows it to more sort of carefully look at the outliers and make sure that they are actually correspond to sort of real biological clusters and not technical noise. So finally, a few words about clustering to think about, because it is a very important step which is part of this analysis, and you will sort of because a lot of the sort of what happens downstream is sort of heavily dependent on how you decided to group your cells. So it's in many projects, this is sort of where you'll be spending a lot of time thinking about which clustering solution is the correct one. And so some points to think about. So one is that in my experience, in general, clustering tends to work really well, even with very sparse data. And sort of intuitively, I think about it as if you only have a small number of cells, then, even if you only have a small number of reads, a small fraction of the genes detected, really most of the information is sort of in the presence of absence of the gene. And that will allow you to do really well, even if you have sort of, say, fewer than 1,000 genes detected per cell. Another thing to keep in mind is that there's a sort of dichotomy between the sort of mathematical and biological considerations. So from a mathematical point of view, the clustering problems sort of very well defined, and we know that there sort of exists a global optimum and so on. It might be very hard to find, but we can sort of through various means, we can sort of convince ourselves that we've reached it. Unfortunately, sort of the biological the mathematical optimum may not sort of correspond to the most appropriate biological solution. So it is important to sort of take both of these aspects into consideration and really think about how you interpret the solution biologically. And for those reasons, perhaps you want to choose something which is sort of suboptimal from the mathematical point of view. And again, sort of related to what I was saying before, that sort of it works. So these methods work quite well, and you can cluster even very large data sets in sort of relatively manageable amount of time. But what you'll typically spend much more time doing is sort of annotating these clusters and understand what they represent. And today I would say that this is really the biggest bottleneck. So next I'm going to talk about some strategies for trying to sort of understand these different clusters and sort of just annotating them. So one concept which is sort of intuitively very easy to understand is marker genes. So marker genes are defined as genes that are highly expressed in one cluster and lowly expressed everywhere else. So this is sort of illustrated here in this heat map. So here on the rows you have different types of genes, and in the columns you have cells which correspond to different clusters. So you can see the diagonal here, which corresponds to the marker genes. They are sort of highly expressed in one cell type and low pretty much everywhere else. So these are typically very useful. If you find those, you can go into the literature and try to understand sort of what each of the different clusters that you identified represent, because there is a lot known already about different markers corresponding to different cell types. Pretty much all clustering packages will get you a list of marker genes for each of your clusters. There are, of course, specialized methods for finding marker genes. One such method is called COMET. And unlike what the most clustering packages do, which is just look at each gene individually, it sort of uses a combinatorial approach to sort of try to find the best set of genes that will represent each cell type. So, of course, there are sort of on the order of 20,000 genes in the human and mouse genome. So looking at all combinations of pairs or triplets is not possible. So what they do is that they have a heuristic, which is quite straightforward. They rank the gene based on their sort of marginal performance, and then they take the top of that list, and then they look at all combinations in the top, and that sort of obviously saves them a lot of computational time, and it seems to be working very well in practice too. So another approach that you can use in order to annotate your clusters is to sort of try to take advantage of other data sets. So in particular, now that there are these sort of various atlas data sets that are starting to come out, what would be really useful is if we could sort of try to leverage what is in there and use that when we try to annotate our newly collected data sets. So this is when you sort of try to sort of essentially map your existing data set to an atlas or to some other existing data set in order to figure out what it best matches. So the first method to do this is called SCMap, and it takes a sort of a very simplistic approach. It essentially it thinks of each of the existing clusters that you had in your atlas or in your reference as a centroid, and then it for each of your new cells from your query data set, it tries to sort of just match it to the nearest centroid. And what it sort of does in order to do this successfully is it uses sort of feature selection. It uses this dropout based methods that I told you about earlier. So based on that, it only looks at a subset of the genes and calculate the distances to these centroids and matches it to the closest one. This type of problem is sort of quite well set up for sort of a lot of other machine learning techniques, and you'll find many other methods out there. One example is single cell nets, which is based on random forests, and it sort of does pretty much the same thing as SCMap does. So all of these, both of these methods and many others are sort of unsupervised methods, and that they trying to learn how to best project them. The data sets. There is, of course, as you know, a lot known about various marker genes and so on for existing cell types. So it would be, of course, useful if we could take advantage of that information when we try to do this projection or mapping. There are methods for doing that. So those I label as supervised projection methods. And one such example is Garnet. And what it does is that you'll have to give it a sort of a hierarchy of the cell types that you think you have in your sample, along with a set of marker genes for each one of those. And then it sort of uses that and looks at the data, and it tries to build a classifier that will tell you which cell type the cells in your queries sample matches. So we talked about visualization before, and it's pretty tricky for these high dimensional data sets. So it will obviously be even trickier if you have two different data sets that you, so one of is your atlas and the other one is your query data set. So one neat way of sort of trying to represent these types of mapping is using these Sankey diagrams. So here I'm showing you one example. So what we have here is sort of on the right, we have the reference, and on the left, we have the query data set. So in both of these cases, the data sets had been annotated beforehand, and then we use the method to map them. So what you can see here is that most of the cells that were labeled beta cells in the, so these are from a pancreas data set, maps to beta cells in the reference data set as well, and a few of them go other places. What's important to point out here is this unassigned category, and this is sort of course not in the reference, but it's something which is very useful to have for your mapping algorithm. So what this really means is that it means that the cells that it found that were given to it and for your query data set do not match anything that was present in the reference. So this is essentially a way of for the algorithm to say sort of, this is something that I've never seen before, and this could be the way that you will discover sort of new cell types that were present in your new sample but not in your reference. So something which is sort of related to these types of questions about marker genes is differential expression. So this is arguably one of the most important applications of bulk RNA-seq to sort of identify the set of genes that are different between two samples. In the single cell world, it's actually more complicated than in the bulk RNA-seq world, because what you typically the groups that we want to compare are not defined a priori, because what we typically do is that we first do unsupervised clustering to identify the groups, and then we start comparing them. And what that means from a sort of statistical point of view is that that means that we'll get more, if we just run the sort of same old differential expression test, we will get more differentially expressed genes because the clusters are, of course, based on defined on differences in gene expression. So that will result in sort of more differentially expressed genes detected. So that's something which is important to keep in mind when you're doing this. So there are lots of methods for doing a differential expression for bulk RNA-seq. There are also lots of methods that have been designed specifically for single-cell RNA-seq. What's interesting in this is in this big benchmarking studies that was done a few years ago, and they tried to compare these. What turned out was rather surprisingly, I think, at least from my point of view, is that one method that performs really, really well, and you can sort of see here by blue meaning that it does well and red meaning that it does poorly, is just the standard Wilcoxon rank test. So this is a non-parametric test, so it's very easy to use and it's very robust, and so it has really become the standard to use in the field these days, and that's what most people, including myself, would recommend that you use for differential expression in single-cell RNA-seq data. one problem that you often have when you're trying to do this differential expression analysis is that you will end up with a lot of different comparisons, especially if you have sort of multiple data sets from different conditions that each have been clustered. So you have multiple groups. Then you can end up with having a very large number of pairwise comparisons, which will be sort of problematic in terms of cognitively for you in order to be able to process, but also perhaps more importantly, from a statistical point of view, that you'll have a lot of multiple testing correction that can sort of result in you losing power. Fortunately, there are methods from statistics to deal with these types of situations. And if you're faced with this type of complex experimental design for doing differential expression, one method to consider is called MuSCAT, which sort of uses a generalized linear model framework in order to try to be able to group samples and in sort of in meaningful way, and it will give you better power. So I think I will stop here. Okay. Yeah, so of course, there are situations where sort of doing this type of hard clustering, finding discrete sets or categories where your cells should fit is not really the most appropriate approach. So in that case, we need to have a different approach, which is called pseudotime analysis. So in pseudotime analysis is sort of typically used when we have some sort of continuum. Most often we think of that as when we have some sort of differentiation process. So when cells sort of start out in one cell type or cell state, and they sort of gradually move or progress into a different stage over time. So the problem with the single-cell RNA-seq assay is that we can only measure each cell once. So because we lyse them and then they can't go on. So what will happen is that we essentially get like a snapshot of what's going on in our sample. So and if we have this type of differentiation process, what will happen is that some of the cells that we profile will be sort of at the earlier stages of this differentiation, and some of them will be closer to the end. But a priori, we don't know where each cell is. So really the goal behind pseudotime is to sort of try to sort of align, essentially project or align the cells in a one-dimensional setting. And then we can sort of, once we've ordered them, then we can look at sort of different properties, such as expression profiles of certain genes, see how they vary, and sort of learn different things about this differentiation process. Doing this is sort of in the one-dimensional setting is sort of relatively straightforward. The much more interesting and of course more complicated situation occurs when you have branching going on. So this happens very frequently when you have two or more different cell types that sort of share a progenitor during development. So I'm showing you here on the left, arguably the most sort of well-studied and famous example, which is the hematopoietic system, where the hematopoietic stem cells will differentiate into various types of these blood cells. So what we need to do, again, now from a sort of mathematical point of view, is to sort of identify, ideally we would like to have an algorithm that sort of automatically identifies these branches, their endpoints, sort of both and their locations, and how many there should be. And this is, of course, much more challenging than doing just ordering them along a single dimension. So there are multiple methods for doing this. A few years ago, at least, there was a review, and I think it turned out that this is pretty much the most popular field within or area within the computational single-cell RNA-seq in terms of the number of algorithms that have been published. So the first method to do this was it's called Monocle from Cole Trapnell's lab. This is old these days. It's not really used, but there is Monocle 2, which is quite popular. So they use a reverse graph embedding strategy. And I mean, very loosely speaking, what they're doing is that they're essentially trying to find a sort of principal graph which is a sort of extension of the idea of a principal curve, which sort of tries to go through capture as many of the points as possible by sort of going through their centroids. So the monocle 2 will sort of automatically try to identify the number of branching points and their locations. So one drawback of not just monocle 2, but most of these strategies or methods for doing this is that they can't really tell you that they're not directed. So they can't tell you where the process starts and which is the starting point and which is the end point. In my opinion, this is not really a major drawback because typically we can sort of have quite a lot of biological knowledge what's going on, and by looking at the profiles of the cells near the different ends, we can usually figure out quite easily which one is the start and which one is the end, or which ones are the end if you have branching. So a different method is called diffusion pseudotime. This is based on the idea of these diffusion maps, which is basically trying to find a sort of a low-dimensional diffusion kernel to fit the data. And it's sort of, you can think of it as it sort of tries to, based on the ideas of random walks, it tries to find the sort of the shortest random walks for connecting these different points. And again, this is a very robust method which seems to perform very well. So one of the these pseudotime methods, they really only work for doing sort of one-dimensional or sort of lower dimensional when you have a few branching points, but they don't really apply to sort of the type of big clustering studies. And if you want to understand how the cells are sort of moving or transitioning in that space, then it gets much more complicated. But fortunately, there's a very clever method called RNA velocity, which really effectively allows you to understand the sort of the direction of each of these points or the cells in this cell state space. So the idea is sort of based on a really clever observation that even though these different assays that we have are sort of, they look at polyadenylated RNA, you will still get some reads from pre-spliced RNAs. So you'll get some reads that correspond to introns and are hence unspliced. And then what you can do is that you can sort of set up this very simple first order differential equation for how the RNA gets transcribed and spliced, and then using this information about this number of spliced and unspliced reads, you can fit the parameters for this ODE, and then given these different parameters, we can infer sort of what is the temporal dynamics of these different genes, and then what we can moreover do is that we can look at the individual cells, and we can sort of place them in this phase diagram, and we can sort of infer for each cell if it seems to be in the process of sort of producing more transcripts, or if it's in the process of where degradation is dominating over transcription, so it's sort of going towards zero, or if it sort of seems to be close to equilibrium. So we can do this for each gene in each cell, and then we can basically calculate a velocity vector that we can project onto the same space, and that really allows us to understand sort of how each cell is sort of moving around in this high dimensional space. So this is really elegant and is really powerful for understanding sort of what kind of transitions you can infer from your data. So what's also really useful is that you can actually you can do even better. You can actually go back into and connect this to real time. So this was done, for example, in this single cell malaria atlas that I told you about. So there we have this life cycle of the malaria parasite, which is as the name implies, it's a cycle. So it makes sense to sort of plot the data in this type of cylindrical fashion. And then by relating this to other external data sets where people have taken sort of bulk sets of these parasites and sort of synchronize them and then profile them at different times, you can sort of identify certain key events, and then you can relate those to real times, and this allows you to understand how this snapshot type of data applies or correlates with the real-time events. So I will stop here before we go on to the next. Right. So this last section, I'm going to be telling you about sort of two other types of applications, which are sort of not directly linked to the expression matrix or the count matrix, but they're nevertheless sort of very useful, and it's important to know about them these days. So one of the consequences of this sort of a huge improvement of single cell technologies, coupled with a sort of continued reduction in sequencing costs, is that people are sort of dreaming up and coming up with more applications than they did before. So one of them is sort of really more human genetics inspired. So we can now start thinking about human sort of variation across the genotypes. So the idea is that you would sort of profile the same cell type from lots of different donors, and then if you also have their genotype, you'll be able to sort of infer which variants are sort of responsible for differences in expression levels across these different donors. So we can really start to identify these expression QTLs. And this will, of course, be very important from a sort of the basic biology point of view in understanding sort of how non-coding variants can impact expression levels, but also sort of in the bigger scheme of things, it will hopefully sort of take us further along the way of sort of personalized medicine for clinical applications. So the first sort of study that looked at QTLs in a single cell data set and in a sort of meaningful level was published in 2018 from the Franke group. So they looked at about 25,000 cells from 45 different donors. So they found, I think it was 379 different cis-eQTLs. Most of these, I think about 330 or so of them, were also found in a sort of equivalent bulk RNA-seq sample. So most of the so you don't find a whole lot new, but there are some interesting features that you gain from the sort of single cell perspective. So one of those is sort of shown here. So with this variant here. So what you can see here is that you have basically different trends with sort of here you have in the CD4 T cells, you have higher expression levels if you're homozygous for C than if you're homozygous for A, whereas for the NK cells, you have the opposite trend. So what this means is that in the bulk RNA-seq, these two things essentially cancel each other out, so you can't see the effect. But in the single cell RNA-seq data, you see that you have these cell type specific QTLs, which is sort of what we were hoping to find. So again, sort of going back to the more sort of pre-processing stage of this, one challenge that you have here is that if you try to if you sequence each donor separately, then you'll have the problem of batch effects. So, but there is, of course, a way to get around that, and that is if you just pool all of your cells together, and then sequence them, then you don't have the problem of batch effects. And then we can use information about the different variants from SNPs in these cells to sort of demultiplex the cells later, and so basically identify which donor each of the cells came from. So, there's major advantages that we get rid of batch effects, but it is also importantly from a cost point of view. So it allows us to sort of run more cells per sample, and which will sort of reduce the overall costs. So the first method to deal with this is called demuxlet, and it requires, in addition to your single-cell RNA-seq data set, it also requires genotype profiles for each of these each of the donors that you have in your sample. So one, you might think that it could be problematic if you have this type of droplet-based data where you only get sort of primarily the, say, the 3' or the 5' end of each gene. But it turns out that the authors here did some analysis, and they were able to show that if you have, say, 64 donors, and in that case, you only need to have sort of 50 informative SNPs in order to be able to desegregate the cells with a high accuracy. So this is indeed sort of well suited for these types of technologies. So in the second generation of these tools, they did away with a very important step here, which is having a known reference. So one such example method is called supercell. So in supercell, you only require single-cell RNA-seq data. You don't require the genotypes. And so what supercell does is that it starts from the original reads, and it remaps the reads using Minimap2 and using sort of settings which are more favorable for being able to do this variant calling. Then it does the variant calling, and then it builds up this cell by variant matrix, which is then uses for clustering. And one advantage of this clustering is that it gives us sort of two pieces of information sort of false out more or less for free, which is very useful. So one is sort of doublet detection, and it's important here to realize that there are sort of different types of doublets. So one is referred to as sort of genetic doublets. So that's when you have two cells from different genotypes. So those are the only ones that we can detect. If we have two cells from the same donor, then we can no longer detect them using this method. Then we have to use the type of methods that I talked about in the previous lecture. So in this space, the doublets come out very nicely because we expect them, each cell to be sort of very near a cluster. But if we have things that are sort of lining up in between clusters, they are very clearly doublets. The other aspect that comes out sort of for free is an estimate of the amount of ambient RNA. And the reason for that is because we know that each allele frequency should be sort of very close to either 0, 0.5, or 1.0. So if we observe sort of reads or alleles that have sort of deviate significantly from those ratios, then we can be pretty sure that is due to ambient RNA. So transcripts having sort of bled into every droplet from outside. And so the method can actually produce what we believe is a very good estimate for the amount of ambient RNA in your sample, which is quite useful. So the second type of method that I want to talk about here is something called CiteSeq. And what CiteSeq does is that it allows you to measure not just the transcriptome, but also some levels of some proteins. So it does this in a very clever way, is that you take antibodies that are sort of specific to certain the proteins that you're interested in. So you typically have a sort of relatively small or a panel of antibodies that you can look at. And then each of those antibodies is tagged with with a nucleotide sequence. And so then after we've done our sort of droplet encapsulation, we can release the tags from the antibody, and then we can sequence those together with all the RNA, and that can give us a good estimate of the protein levels that were found in these cells. So this is, of course, there are sort of limitations. So we need to have a good antibody, and we can only look at proteins that are on the cell surface. But even with these restrictions in mind, this is very, very useful. So one thing that it allows us to do is to sort of use a sort of single RNA-seq sort of in a way that we would sort of use flow cytometry. And as you can sort of see here from this figure from their paper, this seems to work really, really nicely. And one main advantage here is that protein levels tend to be manyfold higher than RNA levels. So for the RNA-seq data, we have all these issues with high sparsity and dropouts. But for the protein data, this is typically not the case, and it tends to be sort of look very nice and Gaussian. So this is a very, very nice feature to have. The other thing that this has been built on is to develop this method called cell hashing. And the idea here is that you can use this for doing demultiplexing. So you can basically add these antibodies with these specific to each of your samples. You add the antibodies with the specific tags, and then you can do the sort of demultiplexing later on. So that again will sort of allow you to get overcome batch effects, and possibly also save some money when you're doing your experiment. This method has been expanded, so they have Excite-seq, where they also incorporate looking at sort of VDJ sequences and gRNA. So you can combine this single-cell RNA-seq with CRISPR perturbations and do the readouts from the same cells and multiple things. So that's quite handy. So yeah, so this really takes me sort of to the end of this roadmap here. And yeah, I think we've sort of covered most of the major steps here and some of the tools that are available for doing this. What's really important to mention is that this is a very fast-moving field. So people are sort of constantly publishing new methods that sort of try to do better for these different steps that I've been discussing. But perhaps more excitingly is that people are also sort of inventing entirely new things that you can do with this data. So for example, 3 years ago, we wouldn't have been talking about RNA velocity or clustering by genotype, but now these are sort of, I would say, sort of quite standard things as part of the sort of standard processing pipeline. So I would expect there to be sort of more development like this in the coming years. So finally, what I should mention is that many of you have already talked some about Seurat, and many of you may have already have heard about it. So this is probably the most widely used and popular method in the whole field. So it's basically a toolkit that sort of will do sort of pretty much most or all of the steps that I've told you about before here. There are methods in Seurat for doing this. So I think Seurat is developed and maintained by the Satija Lab at NYU, and they do a really nice job of they put together really good vignettes and documentations, and they maintain it actively. So I think now they're at version 4.0. So this is a very, very good package to use if you're going to sort of start somewhere. The one drawback, depending on sort of how you feel about your programming languages, is that this is built in R, which I find a bit of an acquired taste. So one alternative is to use ScanPy instead, which was developed by the Theis Lab, and they also have sort of very nice documentation and sort of actively maintaining this, and this does pretty much the same thing as Seurat, but it's based on Python. And I think by most benchmarks, it would be more efficient, faster, and less memory intensive, because that tends to be happening when you compare R to Python. So what I should mention now is that there are sort of when I first started, pretty much everything was done in R. But over the last few years, there has really been a sort of trend that more and more things are being done in Python, and I think that is sort of set to continue. Now, of course, the good news is that there are also sort of interpreters, so it is possible to sort of call R code from within Python and vice versa. So just because your favorite package is not written in the language that you want, you don't have to switch your entire pipeline. Should be good news. So with that, I will just sort of mention there are the sort of what I think are the really hot topics in this field. And again, given how fast the field is moving, you'll probably be hearing about them next year already. So this is sort of multi-omics. I touched a bit on that, but there are sort of other types of omics that people will be comparing. And I think in particular, you'll see a lot of sort of RNA plus ATAC combined. There are already several protocols out for that, and 10x recently released a kit for that, which will mean that it will have much greater uptake. And then there's sort of spatial methods where it basically comes down to image analysis, where you sort of look at a section of a tissue, and then you can put it into spatial context where each cell that expresses variant transcripts are found. So I think with that, this is the last slide. Much of this is based on some material that me and my group put together. You have the link over there. Fortunately, it's a little bit outdated, but you'll still find the sort of the overall framework should be okay. There are a lot of exercises in there, so if you want to sort of try out yourself to learn how to do these things, and with answers being available, you can go there. And then I also put a link to a very nice a review which sort of goes over these steps and tells you what they think are the best approaches to use. So with that, I will stop sharing, and I will stop the recording.